Hello friends, welcome to some biology class. Friends, today we shall discuss about lymphoid organs. The immune system consists of many organs and tissues which are found throughout the body. These organs, called as lymphoid organs, can be classified functionally into two main groups such as primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs such as bone marrow, thymus and bursa fabriceus provide the appropriate microenvironments for development and maturation of lymphocytes. In these organs, the lymphoid stem cells proliferate, differentiate and mature to become immunocompetent. The mature lymphocytes of primary lymphoid organs migrate through blood and lymph into the secondary lymphoid organs where they are exposed to different antigens. As a result, they become functional. Examples of secondary lymphoid organs are spleen, lymph nodes and mucosa-associated lymphoid tissue. In secondary lymphoid organs, the mature lymphocytes interact effectively with antigens. Now we shall discuss about bone marrow, thymus and bursa fabriceus, which are all primary lymphoid organs. Bone marrow is a soft tissue located within the cavities of bones. Hematopoietic stem cells develop in the bone marrow. From these stem cells, all types of blood cells such as red blood cells and white blood cells, which include granulocytes, lymphocytes and monocytes, are all derived. In addition, thrombocytes, also called as platelets, are derived from these stem cells. In bone marrow, B lymphocytes proliferate, differentiate and mature to become immunocompetent. Another primary lymphoid organ is thymus. In human being, thymus is bilobed and is located just behind the top of the sternum below the thyroid gland. Thymus is enclosed by a fibrous capsule. Each lobe is organized into many lobules which are separated from one another by septa called trabeculae. Each lobule is organized into an outer cortex and inner medulla. The main function of thymus is proliferation, differentiation and maturation of T lymphocytes. Bursa fabriceus is another primary lymphoid organ which is exclusively present in birds. In birds, stem cells develop in yolk sac, liver and bone marrow, from which some of the stem cells migrate to bursa fabriceus where they proliferate, differentiate and mature to become immunocompetent B lymphocytes. Bursa fabriceus is a sac-like lymphoepithelial structure which remain attached to the dorsal side of cloaca. Secondary lymphoid organs trap antigens from defined tissues and vascular space. In these organs, mature lymphocytes interact effectively with antigens and brings about humoral and cell-mediated immune responses. These organs are small and poorly developed at birth. They grow progressively with age. Lymph nodes, spleen and mucosa-associated lymphoid tissue are examples of secondary lymphoid organs. Lymph nodes are complex, cellular, spherical or ovoid structures present along the lymphatic ducts. This is a lymphatic ducts and these are the lymph nodes. In human being, lymph nodes are bean-shaped. Several efferent lymphatic carry lymph into the nodes, whereas from each node lymph is carried out by a single efferent lymphatic. Lymph nodes are covered by a capsule. This capsule penetrates into the interior here and there by forming septa called trabeculae. The lymph nodes are organized into three distinct regions. Outer is called cortex, middle paracortex, and the inner zone is medulla. Cortex contains B cells, paracortex has T cells, whereas medulla contains both B and T cells. The main function of lymph nodes is to filter lymph. They are important centers of phagocytosis and are responsible for development and initiation of humoral and cell-mediated immune responses. Another secondary lymphoid organ is spleen. It is the second largest lymphoid organ in our body. Due to the presence of large amount of blood, spleen is deep red in color and it has direct communication with the main arterial circulation. Like lymph nodes, spleen is also protected by a fibrous capsule. The capsule penetrates into the interior here and there and forms septa called trabeculae. Spleen is distinguished into two different regions, red pulp and white pulp. Red pulp contains many blood-filled sinusoids in which phagocytes and plasma cells are found. The main function of red pulp is destruction of old and dead red blood cells and other cellular debris. The other region of the spleen is white pulp, which contains the lymphoid tissue. A major part of the lymphoid tissue is arranged around the central arteriole, forming periarteriolar lymphoid cells. Spleen trap blood-borne antigens, so it acts as a filter of blood. It is responsible for development and initiation of humoral and cell-mediated immune responses. It produces all types of blood cells during fetal life. In addition, spleen also produces opsonins, properdins, and toxins. It is concerned with release of neutrophils following myocardial infarctions. Spleen also stores red blood cells, lymphocytes, and other formed elements. Mucosa-associated lymphoid tissue, also known as MALT, is the secondary lymphoid tissue without capsules. These tissues are found dispersed in the mucosal layer of the alimentary canal, respiratory, and the genitourinary tract. Mucosa-associated lymphoid tissue is non-encapsulated. Pairs, patches, and tonsils are the good examples of MALT that are present in the mucosal layer of the alimentary canal. Hence, they are also known as gut-associated lymphoid tissues. So friends, this is all about various lymphoid organs and tissues that are present in our body. For more updates, please subscribe my YouTube channel, Sam Biology Class.